So this document is actually in Google Classroom for you guys, so you don't have to write this stuff down, but you still may want to make some notes as we go along. Okay, but that document is in the class work tab. And so if you go to period 1 here, okay, class work, and it's in class materials here at the bottom. Okay, and right, it's lab report expectations right here. All right, so you can kind of follow along if you want to open that one up. Okay, follow along with that on your phone, okay, or whatever. But you will want to write a few things down as we go along here. All right, so the kind of format here, the way that lab reports are going to work for you. Anytime we do a lab, I will give you a template for that particular lab report. Okay, so on well, tomorrow actually, right? You'll get a file in Google Classroom, okay, through an assignment that's U1 lesson 5 properties of compounds lab, and it'll say your name beside it. Okay, so that'll be the template that you will use. It will be partially filled in already. Okay, so it'll say problem, and there'll be a blank, and you'll have to fill in the problem. It'll say hypothesis underneath that, and there'll be a blank, and you'll have to fill in your hypothesis. Okay, it'll say your variables, your design, and ask you to identify and explain your variables, manipulated, responding, controlled. Okay, there'll be a materials list. Okay, that'll be filled in already. I'll have the materials in there. Okay, then there'll be a procedure. For this lab, you're actually going to write up the procedure. That's what we're going to be working on tomorrow. After the procedure, there'll be some charts for observations, okay, for you to record stuff in, and then there'll be an analysis. Okay, you'll have to fill that in, answer those questions. It'll say conclusion, you'll have to write your conclusion, and it'll say underneath that evaluation, that's where you fill in the sources of error. We're going to talk about all that stuff today, but you'll get a template that has all of that built. Okay, so that'll be the format that you will be expected to follow. Right? I'm going to tell you today, here are the expectations for each part of that lab report. Right? So if you want to do well on your lab reports, you want to follow these expectations. Okay? Don't be afraid to ask questions as we go. All right, the first thing that's going to be on your lab report is the problem. Okay? And the problem is going to be a question in your own words. Right? So I'll already have a problem like written out. What you'll have to do is write it in your own words. So maybe our lab is on the effect of light on plant growth, something pretty simple. Okay, so your very simple problem for that could be, what is the effect of light on plant growth? That would be your problem. Okay, it's stated in the form of a question. It's what we're investigating. All right, so problem can be pretty simple. Sometimes it can be more complex as well. Okay, this is just an example, so it would be the simplest thing we can think of. Okay, after the problem is going to be the design. And, oh, sorry, Matisse, question? It's in the classwork tab, and it's under class materials. If you click on class materials, it's one of the files there. All right, after this will be the design, and this is where you identify your variables. Okay? All experiments have a manipulated variable. That's the thing being changed by the experimenter. Okay? A responding variable, the thing that the experimenter is observing, measuring, or watching for changes in. Okay? And 3 control variables. Controlled variables are things that could affect the experiment if they were not kept controlled or the same throughout the experiment. So if I'm doing a lab on the effect of light on plant growth, my manipulated variable is going to be what? I want to study the effect that light has on plant growth.
The amount of light.
The amount of light, exactly. Okay, that's going to be my manipulated variable. It's the thing that I, as the experimenter, am going to change, okay, in order to see what its effect is on the growth of a plant. Which means then that my responding variable is going to be what? What's going to respond to the amount of light the plant gets? The growth. Okay. Now, when we're identifying these variables, we don't just say my manipulated variable is light and be done with it. No, we have to explain. In that explanation, why is it light? Okay, why is light the manipulated variable? How is it being manipulated? Okay, what is it you're doing, okay, to the light? Okay, for your responding variable, very similar. I identify my responding variable and I explain how it's responding and how it's being measured. Are there a lot of different ways to measure plant growth? Yeah, okay, I mean, the simplest idea would be measure how tall the plant gets, but what if this is a plant that crawls? Okay, or what if this is a plant that has some kind of weird spreading growth pattern that's really hard to measure? Okay, then maybe I'm going to measure its mass instead. Okay, or maybe I'm just going to take qualitative observations and take pictures of it as it grows. Maybe I'm going to measure how much fruit it produces when it's mature, something like that. I would have to explain, this is my responding variable, here's how it's responding, and how I'm going to measure that. Okay, all of that needs to go in the explanation. Okay, for your controlled variables, those are the things that could affect the growth of a plant other than light. Okay, what are some other things that could affect the growth of a plant? What do you have to do to a plant on a regular basis? Water it, yeah. Okay, so I should make sure that all the plants I'm testing, because I'm going to have maybe some that get no light, some that get 24 hours of light, and probably some in the middle that get, maybe 6, 12, 18, 24 hours. Okay, I'm going to have some various amounts of light there. But I need to make sure all those plants get the same amount of water. Otherwise, I won't know whether it was the amount of light or the amount of water that they got that made them grow. Okay, so I need to make sure they all get the same amount of water. What else could affect the growth of a plant that I would want to control? Sure. Temperature, absolutely. Okay, I don't want to have one plant that's growing outside right now because it won't be growing. Okay, and another plant that's in a greenhouse. Okay, that's hardly a fair comparison. I would want them all in the same room, probably in their own box with a light that I could turn on and off. Okay, that way I'm making sure temperature stays the same for all of them. I can water them all easily and give them the same amount of water. Okay, what else should I control? Something else that might affect their growth that I could make sure was the same for all of them. What do they typically grow in? Yeah, size, like the pot they grow in, the soil I put in there, okay? Even that they're all the same kind of plant, okay? Like I know these things would be kind of like common sense, like you wouldn't compare a carrot and a redwood tree and and a tomato plant, like they're not the same kind of plant. It's not hardly a fair experiment, okay? But these are the things that need to be listed. These are controlled variables, okay? These have to be the same, and they have to be the same for this reason. So we're identifying our controlled variables, we're explaining how they're being controlled and why they're being controlled. Okay? This has to be controlled because plants need water to grow, and if they get different amounts of water, they'll grow differently. I want them all to get the same amount of water. Okay? I want plants to all be at the same temperature because temperature can affect the rate of growth of a plant. Okay? Therefore, all the plants will be kept in a box at 20 degrees Celsius, and so on. You just explain, here's a controlled variable, how it's being controlled, and the effect it would have if it wasn't. Right? So, when we're writing out our lab report, we would have, okay, our variables. Okay, so our manipulated variable, that's the part of the experiment changed on purpose by the experimenter, that is what's being tested. Okay, in this case, it's the amount of light given to plants because all subjects will receive different amounts of light, and light powers photosynthesis. So I can just stretch it. Right, so I've identified the variable and I've said how it's being manipulated. They're all getting different amounts of light, okay, and light does this. Okay, so I've said lots of stuff in there. Okay. What I typically see when people do this wrong is this. Manipulated variable, light, and nothing else, okay? Or manipulated variable, light, because we changed it. Okay. Is that a very thorough explanation as to why it's the manipulated variable? It's not, okay? So this is like a grade 10 or high school level explanation, okay? In grade 8 or 9, maybe because we changed it was good enough, but it isn't now, okay? We need to have a thorough explanation as to why this is the manipulated variable. All right, then for our responding variable, okay, we talked about that already, okay? Whatever changes as a result of the manipulations made by the experimenter. So for this one, the growth of the plants, because getting more light will have more energy for growth, okay? And then I would probably also write in there how I was going to measure that, okay? Probably taking the mass of the plant each week to see how much of it grew rather than try to measure it, because all plants are going to grow a little bit differently. Okay, and then lastly, and these are usually the toughest ones, are the controlled variables. Right, so that's what we had just talked about there. Okay, so there are other factors that could affect the outcome of the experiment if left unchecked. Right, so same species of plant, because different plants grow at different rates. Okay, same amount of water, because water is an important factor in plant growth, so each plant needs the same amount of water, so it can't affect the growth rate of the plant. Same soil, because plants get nutrients from soil, and different soils hold water differently. Keeping them the same ensures it cannot affect the experiment. Same temperature, plants typically slow their growth in extreme heat or cold. Choosing an effective temperature for all plants ensures it does not affect the experiment. Okay, I've identified these are things that could affect my experiment. I'm going to control them by doing this. I explain each one separately. Okay, the most common thing I see for people doing this one wrong is they say, here are the controlled variables, we kept them all the same. Great, but that's not a 5 out of 5 explanation. Okay, everybody with me there? All right, so the design part of your lab is worth 5 marks. Okay, so this part here, this is worth 5 marks. Okay, you identify the variables correctly, okay, that's 2 or 3 out of 5. Explain them all in detail, that's 5 out of 5. Okay, so it's not just identifying, it's also explaining why. Okay, questions on the design part? Now, typically on a unit exam, I'll have a few multiple choice questions about experimental design. So I might have something like, a lab is doing a test on bulletproofing glass for use in banks. Okay, their experiment is testing different thicknesses of plexiglass against the penetration of a bullet. Okay. What would be the manipulated variable in that experiment? The thickness of the plexiglass. Okay. What would be the responding variable in that experiment? What happens to the glass? Okay, that would be my responding variable. How far does the bullet penetrate into the glass? Okay, that would be the responding variable. That's what we would be measuring. Okay, or whether the glass just plain shatters. Okay. And then controlled variables. Look, there's some things. If I'm running this test, okay, I've got 5 different sheets of plexiglass. Okay, what kind of things do I want to do the same each time I test the glass?
Under the size of the bullet, the same place on plexiglass, and you want to test the velocity of the empty rectangle.
Yeah. Okay. Basically, I want to fire at each piece of plexiglass in the same way. Okay. So I would have them all lined up, and I would have a line that the person holding the gun would stand behind. Okay. So that they'd be the same distance from each piece of plexiglass, because the further a bullet travels, the slower it goes. Okay. I would probably shoot the same gun with the same ammunition. Okay. It would hardly be fair, right, to take, like a little 22, okay, and shoot it at one piece of plexiglass, and then take, like a Desert Eagle 50 cal and shoot it at the next one. Okay, it's hardly a fair comparison. Okay, one's got a gigantic high-speed bullet, and the other's got a tiny little thing. Okay, so we wouldn't want to do it. We'd have to test each one the same. Same shooter, same weapon, same ammunition, same distance from the glass, same temperature. Okay, all of that kind of stuff would all have to be kept the same in a controlled experiment. Okay, does that make sense? So you'd often get a question that would be something like that. Maybe it would ask you, what's the manipulated variable? Or what's the responding variable? Okay, things like that. Okay, next thing in a proper lab report is your hypothesis. Okay, your hypothesis is not a guess. Okay, I know that probably up until now you've been told that. A hypothesis is an educated guess. No, it's not. Okay, a hypothesis is a statement. It is a statement that sets you up to be right or wrong. Okay? A scientific hypothesis is not like politics. You cannot sit on the fence in science. You have to set it up so that your hypothesis will either be proven correct or proven incorrect. Okay? So, if I'm making up a hypothesis for my plant growth experiment, okay, I have to say unequivocally what I think the effect of light is going to be. Okay, so I would say, you know, that probably light would cause plants to grow faster, but I would say it in this way. A hypothesis always contains three words: if, and, then. It is a deductive statement. If this is true, and this is done, or this experiment is performed, then the results will be this. Okay? Here's what I think is the cause of this problem. This is how I'm going to test it, and here are my results if I'm right. Okay? That's where you're setting yourself up to be either right or wrong. Okay? I think this is the cause, this is how I'm going to test it, this will be the result. Okay? So if this is true, and I do this, then this will happen. Okay? It's a hypothetical deductive statement. That's where hypothesis comes from. All right, so an if, and then statement that communicates what the relationship is between the manipulated and responding variable. That's the if part. Okay? So for this experiment, the if part would talk about the effect of light. Okay? If light has a positive effect on plant growth, or if plants need light to grow. Okay? Something along those lines. I'm stating here's the relationship between light and plant growth, manipulated and responding. Okay? Briefly outline the experiment. That's the and part. Okay, so the and part of my experiment would be something like this, just a brief description. And 6 plants, all receiving different amount of light, are tested over a 6-week period, okay, or something like that. Okay, and then outlines your expected results. That's the then part. Okay, for me, the then part would go something like this. Then the plants receiving the most light will show the most growth. Okay, so I've said light makes plants grow. Going to test it with 6 plants. Plants getting the most light will grow. Okay, that's what I'm saying. Okay, I've set it out. I'm either going to be right or I'm going to be wrong. There's not going to be any in the middle. Okay? So it would look something like this. Okay. All right. If plants require light for growth, so there's my if part. That's the relationship. And 5 identical plants are given different amounts of light. There's my and part. Then the plants that receive the most light will show the most growth. Done. That's a pretty simple hypothesis. Okay, because it's a pretty simple experiment. Okay? If an experiment is a little more complex, then your hypothesis is going to be a little bit longer when you're explaining the if part. Okay, when you're explaining that relationship. This is a pretty simple relationship. This causes this, and we're done. Okay. All right. Everybody okay with how that works? Okay. So, like we said here, the if part of your hypothesis, okay, is the cause or the relationship being investigated and describes the experiment, then what to expect if you are. That's how a hypothesis goes. It will always be an if and then statement. Okay, does that sound familiar from junior high? I know if you had Miss Ferretti, I know that her experiments all set up that way. My kid keeps bringing home lab experiments, and they even say, make an if-then statement. It's perfect. Okay? So that's what they should look at. All right. Next part is your materials. Okay? Sorry, it doesn't say that in here, but the next part would be materials, but that would already be put in there for you. Okay? The materials are always going to be part of the template. You're never going to have to decide what materials you're going to have to use. That will already be put in. Okay, your procedure. Detailed step-by-step explicit instructions for preparing and performing the experiment. Okay, by explicit I don't mean it's full of 4-letter words. Okay, not like that. I mean it is clear, outlined, like there's leaving no doubt what to do. Okay, never make assumptions when you're designing a procedure. Write a procedure as though the dumbest person you know is going to have to follow it. And you know they'll screw it up if you don't tell them everything. Okay? Like when my wife and I do any construction, she has to tell me every little thing or I'll screw it up. Okay? She has to write a procedure for the dumbest person she knows. Okay? So you have to write your procedure, okay, like that. Okay? Don't assume they know anything about what they're doing. Assume they're going to screw it up if you don't tell them. That's how detailed or explicit it needs to be. Now, don't take offense, but I'm going to write most of the procedures for you guys, and I'm not writing them because I think you're dumb. Okay, don't think that. But they are explicit, so you follow them, you can't screw it up. Okay. All right. Then you're going to have observations, okay, chart, graphs, or any other data, okay, including anecdotal notes, okay, that are acquired during the experiment. Okay, so sometimes that's going to be just, the solution turned green. Okay, that's a qualitative observation. Other times it could be something like the cart traveled 5 meters after leaving the ramp. Okay, that's something you would have measured with a measuring tape. That's a quantitative observation. Both are equally valid, right? But observations can come in different types. Okay, anecdotal notes, that would be something like if you were doing a psychological study. Okay, like after 5 sessions of intensive therapy, the participant in this study was better able to connect with their feelings. Okay, something like that would be an anecdotal type of record or observation. Okay, everybody all right with that? Yeah. Once you have your observations, now you have to compare them to your hypothesis. We call that analysis. Okay? Most of the time for you guys, that's just going to be some questions that you have to answer. Okay? For this lab we're doing on Wednesday, it's going to be, all right, figure out what A is, figure out what B is, figure out what C is. That's going to be your analysis. You have these observations that said that substance A was a clear liquid with no odor, okay, that had a pH of 7 and didn't conduct electricity. What was it? Water. Okay? That's analysis. I took the observations I had and I used them to do something. I used it to identify what that material was. Okay, so review and comparisons of data. Most of the time there's just going to be answering questions or performing calculations, and we want detailed explanatory answers. Right, after that is your conclusion. Right, so in your conclusion, the first thing you do is copy and paste your hypothesis. Okay, so just go right back up to the top of the file, highlight your hypothesis, copy, paste. Okay. Then you need to explain to me whether you can accept that hypothesis or reject that hypothesis. So if we go back to our plant lab, okay, let's say that when I performed the plant lab, the observations were as follows. The plant receiving no light died. The plant receiving 6 hours of light grew a small amount. Let's say it gained 100 grams in mass. Okay? The plant receiving 12 hours of light gained 400 grams of mass. The plant receiving 18 hours of light also gained 400 grams of mass. The plant receiving 24 hours of light died. Okay, is my hypothesis acceptable, or do I have to reject it? Because my hypothesis said the plants receiving the most light will show the most growth. Did that happen? No, it didn't. Okay? It looked like it was going to until the plant that received 24 hours of sunlight died. Everything was going great right up until that point. Okay? So, does it mean my hypothesis is 100% wrong? No, I still can't accept it. Okay? It was still wrong, but there was some accuracy to it to a point. That's what I explain in my conclusion. Right? So I would have my hypothesis written there, and then I would say this hypothesis must be rejected because plants receiving the most light, 24 hours, died. Okay? However, there was a pattern of increased growth with increased light up until about 18 hours. Okay? After 18 hours, plants showed degradation. They didn't survive. Okay? Future research should try to find what the ideal amount of light in a day is for plants. That's my conclusion. Okay? I wasn't right, but this is a pattern that I saw. Okay? Further study is needed for this pattern. Is it okay to reject your hypothesis? Is it okay for your hypothesis to be wrong? Yes. You are not going to lose marks on a lab report because you have to reject your hypothesis. Okay? People always think that, and they go back and they're editing, like they're like, oh shoot, that didn't work out. I don't want my hypothesis to be wrong. So they just go back and change their hypothesis. Don't do that. Okay? It's fine. Most of the time, hypotheses are wrong in real science. That's how we learn. Okay? So it's perfectly fine if your hypothesis ends up being rejected at the end. As long as you have a good explanation for why, you're fine. Okay? So don't be afraid to say at the end, this conclusion must be rejected on these grounds. Okay? That is perfectly fine. All right, so the big part of the conclusion is explaining why it's wrong. Okay? If you just say, or right, in a lot of cases as well. Okay? So if you just say, here's my hypothesis, I was right. Oh, goody for you. Okay? It's always nice to be right, but why? How do you know? What told you? Okay? Why did you accept this hypothesis? You can't just say I was right. Okay? You have to explain why. There has to be something there to explain. Everyone follow me? That explanation usually includes some mention of your observations and your analysis. When I was talking about why I would reject my hypothesis, I talked about my observations. Okay? I said the plant that got 24 hours of light died. Okay? But I also saw that plants, as they got more, up to about 18 hours, did better. Okay? Why was that? I don't know. I didn't study that part. Okay? That's for future research. All right. For your evaluation, that's the last part, you identify 2 sources of error within the experiment and how you could fix them. All right, so maybe for this experiment, I found that during the experiment, the plants that got the most light also had their temperature go up a little bit. Okay? Maybe it's just the nature of the light that I was using. Okay? But temperature was not always perfectly controlled in this experiment, so in a future experiment, I may need to have a fan of some kind that's going to circulate the air in all of the boxes to ensure the temperature does remain the same, especially for those plants receiving more light. Okay, it's a source of error. I wasn't aware that it was going to happen. There wasn't really anything I could do about it once the experiment started, but I'm making a note of it. Okay, how would I fix it? Air circulation the next time. Okay, it could be anything. Okay? You're looking for 2 sources of error and ways to fix them. Okay, maybe a second source of error could have been something as simple as some of the seeds did not germinate properly. In the future, maybe more reliable seeds would be used, or only mature plants would be used to start the experiment. Okay, then I'd be sure I don't have a problem with bad seeds or something like that. Okay, everybody follow me there? So sources of error and how to fix them. You are not going to hurt my feelings by identifying sources of error. Every lab is designed to have some. Okay, I want you to find them. Right? You don't have to say to me, Mr. Carey, you designed a really crappy experiment. Here's all the things that were wrong. You're not going to hurt my feelings. Okay, there's going to be things that are wrong. Right? They're meant to be there. All right, everybody kind of follow what we're looking for here. Question?
Do we have to memorize this stuff by our lab through?
No, because you always have this.
Okay.
Yeah. Yeah, and your template's going to have like just the bold stuff, the bold underline stuff will be on there. But yes, you always have this. And if you have questions, guys, when you're putting a lab report together, come in and ask me. Okay, if you're in help, come in and ask me. Okay? I'm always happy to look over a lab if you want me to look it over when you think it's finished. Okay, so if you're done a couple of days early, then pop in some Mr. Gary can read over my lab. No problem. Okay, I'll look it over and give you some tips. Okay, or maybe I just say it's perfect, don't change the name. That happens too. All right. So when we start this pre-lab stuff tomorrow, this first one, I'm really gonna make it easy for you. I'm gonna lay most of it out for you. Okay? Basically, you just have to be paying attention, writing stuff down, so you can kind of get the hang of it. Okay? Now, things that we're gonna have to test in our lab tomorrow, or no, sorry, on Wednesday. Okay. So first test we're gonna want to run. You're gonna need to write this one down. Okay. So this is gonna be part of the procedure you're gonna design tomorrow. Okay. The first test that we want to run is our observable properties test. Okay, so by observable properties, I mean basically all those things that you can observe about the material without doing anything to it. Okay, so what would some of those things be? So samples are all on the front desk. What can I just go up and observe? Color. What else?
Oh, smell.
Odor. What else?
If it's solid or liquid.
State, solid, liquid, or gas. There won't be any gases. They're really hard to keep in a beaker. They just tend to float away. Okay? So there's no gases for you to identify. Okay? But is it solid or liquid? That's important. Okay? If it's a solid, is there some stuff I can record about the nature of it?
It's like, I forget, like the vintage, like temperature solid or butter.
Right, if it's like butter, or if it's crystalline like salt, okay, or maybe if it's molten, right, so kind of like, you know, starting to almost sweat and become like liquid, okay, things like that, okay, those are important. That's the nature of the material, okay. All right, so all of those things are important, okay, observable properties. So color, certainly, okay, the state of matter, solid, liquid, or gas, nature of the matter, crystal, okay, if it's a liquid, is it clear or cloudy, okay, things like that. Odor test, waft the material. When we do the lab on Wednesday, okay, you're going to be using some of the equipment that you can see up here. Right? One of the things you're going to be using is what we call a depression plate. Okay? It's called the depression not because it's sad, okay, but because it's got these pits in it called depressions. Okay? That's where you're going to be putting your unknown materials. Right? Now, each one of these only has 6, so you're going to have to do this twice. Okay? So you're going to put the first 6 in, run all your tests, grab the next 6, run all your tests. All right? Everybody with me so far? Okay? And you can label them from the bottom if you need to, okay, or you can label up here on the top. So we'll have some permanent markers in there, okay, that we can use to label the plates so you know this one's A, this one's B, and you don't get them mixed up. All right. Now, once I've got 6 unknowns in here, do I want to be doing a wafting test? What good is it going to do me, right? I'm going to do this, I'm going to smell them all at the same time. Okay. To do the wafting test, you actually want to waft the sample at the front. Okay. Just pick up the beaker and waft it at the front. Okay, that's the best way to do that because it's a larger sample and the smell will be much more obvious that way. Okay, all the other tests obviously can be performed in here. Okay, so we're looking, like I said, state of matter, color, nature of matter, okay, all of that kind of stuff. Now, once we've done that, the next thing we want to do is a solubility test. To do a solubility test, all we do is add water. Okay, you'll have little wash bottles that are full of distilled water. Okay, you'll simply squeeze them, get some water to go in there, and while you're adding the water, you'll be using this thing. I don't know if you've seen one of these before. It's called a thermometer. You probably call it a thermometer. Okay, so you're going to hold it by this little red rubber piece here. Okay, why? Why wouldn't you hold it like this?
Is that good for the temperature?
Exactly, it's going to read the heat from your hand if you hold it like this. Okay, hold it by the little rubber thing. If yours doesn't have a little rubber thing, okay, hold it by the tip as much as possible. Okay, and you can use it to kind of stir a little bit. Okay, now, if there's a big change in temperature while you're dissolving, is that a useful observation?
Yes.
Yes. Okay, during a solubility test, if there's a change in temperature, it actually means that you're usually dissolving a strong acid or a strong base. Okay, there's also one unknown that's going to get colder. Okay, I didn't see many people write down, so I just gave you 3 really big important things to watch for on Wednesday. If it gets hotter, you're typically talking about a really strong acid or a really strong base being dissolved. You will also have one material that will get colder. How many people have ever used one of those instant cold packs where you pop the little bubble and shake it and then it gets cold? You know, you sprain your ankle, right? You just pop that a little bit. Okay, inside of there, inside the bubble is water, okay? But the crystals that you can hear inside are ammonium nitrate. That's the cold one. Okay? I'm telling you that because that's one of the ones that basically would be really hard to know otherwise. The one defining characteristic of it is that it is an ionic compound, but when it dissolves in water, it gets colder. Okay, so that's our solubility test. Okay, so when you're dissolving it, okay, you're watching for temperature changes, you're watching for color changes. Okay, so if you've got blue crystals, you put them in water, and the solution turns blue. Okay, that's important. Okay, or it turns green, that's important. Okay, and if there's any change in odor. Spoiler alert: there really shouldn't be any change in odor for anything. Okay. Okay, once we've observed whether they're soluble or not, and we've recorded these things, okay, if it dissolves easily, then you can say it's soluble, okay? If there's a change in temperature, okay, you record that. If you're stirring away and this stuff just keeps floating there and doing nothing, not dissolving, then you write that it's not soluble, okay? If you're putting two liquids together, how will you know if it's soluble or not? Can a liquid dissolve in another liquid? Yeah, it can, for sure. They just disappear, okay? Now, if I mix oil and water, that's two liquids. Do they dissolve in each other? No, one floats on top of the other. If you see that, okay, that's not soluble, right? But if I pour a liquid in and I can't see two separate parts, it's soluble. Right, so those are the signs of something being soluble. So if I'm stirring something and I'm seeing it get, it's obviously the solution is changing color, or the material is shrinking or disappearing or whatever, those are all signs that it's soluble. But if I'm stirring and stirring and stirring and nothing's happening, then it's not. Okay, but you do have to watch it carefully. Remember, we don't have a whole lot of water in here, so it's possible that even something that's quite soluble isn't going to completely dissolve in this small amount of water. But if I'm stirring it and I'm seeing that, if it's crystals, the crystals are shrinking or their shape is changing, stuff like that, those are all signs that it is soluble. Okay, those are the things I want to watch for. Okay. All right, once I'm done that test, I now have a whole bunch of solutions. What can I test about them? This is really useful if I want to tell whether it's ionic or molecular. No, I would have tested that in the observable properties. Give you a hint. I would want to use this thing. It has a battery on it and 2 electrodes. What might it test?
It's like conductivity.
It's conductivity. Okay, this is the conductivity tester. Very simple. Okay, I put it in the solution I create and I press the button on the side. Okay, the red light indicating it's working comes on, and then it'll show me on a scale of 1 to 10, okay, how conductive that material is. Okay, like we said before, 7 to 10, it's ionic. 4 or less, it's molecular. You get something in the middle, try it again. Okay, make a new solution and try it again. It probably got contaminated. Okay, you shouldn't get too much that's in the middle. Okay, you should get things that are obviously non-conductive and things that are obviously quite conductive. All right, so the conductivity test is done after the solubility test, because then we don't waste anything, right? We've made these solutions and now we test whether they're conductive or not. Okay, so that would be your next one, conductivity test. Right, so you just look for the lights, okay? And rinse the tester with distilled water, not tap water, in between tests. Why? What does tap water have in it? You guys should really know what's in your tap water. They put something in it to help your teeth.
Is it iron?
Okay, there could be some iron, especially if you have a well. If you have a well, you typically might have more iron. There's not too much iron in Okotoks town water, okay? But if you have a well, you could have some iron in it. Okay, what else? What do they put in water to keep your teeth healthy? The dentist puts it on your teeth too. It starts with F. Fluoride, yeah, okay, there's fluoride in water. There's lots of calcium, okay, in Okotoks water. That's what makes our water so hard. Okay, you might have a water softener at home, okay, and helps take the calcium ions out. There's ions in tap water, okay. If I rinse this with tap water, then I risk putting ions into my next solution that shouldn't be there, and I'll get a false reading. Okay, so I want to wash this with distilled water between, because I know that just cleans it off and doesn't put any ions in. Okay, everybody follow me there? Okay, so we always want to rinse this in between and the thermometer in between each solubility test as well. Give it a squirt of the distilled water, okay, to make sure that it's clean, and probably dry it off with a paper towel between each one as well. All right, and then your last test is going to tell us whether you've got something that's an acid or a base, and that's your pH test. The pH test is pretty straightforward. Okay, you're gonna have these little tubes. Okay. Inside the tubes are little strips of pH paper. On the outside of the tubes is a color chart, okay, telling you what color corresponds to what number on the pH scale. Okay, so if it's super red, it's a 1. If it's really dark blue to black, it's 12 or higher. Okay, anything in the middle is going to have some range, you know, from red to blue. Okay, 7 is typically pretty yellow. So you can probably use one strip for 2 different materials. Just take it off and rip it in half. All you have to do is dip it in and quickly compare it to the chart, and then throw it in the garbage. Okay, so that's your pH test. Okay, and that will help you to determine which of your ionic compounds are the strong acid and the strong base. So basically, which one's hydrochloric acid and which one is sodium hydroxide, because those are 2 of your compounds. All right. That sort of makes sense? We'll go into more detail about that tomorrow when you're designing your procedure and stuff. Okay, so tomorrow we'll have the Chromebooks. Okay, we'll be essentially doing the problem, the variables, the hypothesis, and the procedure. Okay, talking about what kind of observations we want to record the next day. Okay, so you'll have all of class tomorrow after your quiz to work on that stuff. Okay, questions on any of that? All right, so I will post the quiz after school today, so make sure you check that out and give it a try. All right. Okay, if I can get everybody to pack up and clean off the top of their desks, I will come around with the spray and get everything cleaned up here.